Hi students, now we are discussing the chapter Heredity and Evolution. In our previous class we discussed about monohybrid cross. Today we are going to discuss about dihybrid cross. A cross between two plants having two pairs of contrasting characters is called dihybrid cross. A cross between two plants having two pairs of contrasting characters is called dihybrid cross. Here you can see the dihybrid cross. Here the parents, one is male and the other one is female, and you can see the phenotypes for the parents are round green seeded plant is crossed with wrinkled yellow seeded plant. The genotype here you can see in pea plants, as you know, round seeds are dominant over yellow wrinkled seeds. Round seeds are dominant over wrinkled seeds. That's why for the dominant character we used to represent with capital letter R, and for wrinkled character we used to represent it with small letter r. And green seeds are recessive character. Yellow seeds are the dominant character in pea plant or Pisum sativum. That's why for yellow seeds we used to represent that character by using capital letter Y, and for green seeds we will use small letter y. Then we can write the genotype of the parents as capital R, small y into small r, capital Y. As you know that here only one type of gamete is formed from male parent or from the round green seeded plant, and one type of gamete is produced from the wrinkled yellow seeded plant. Here the gamete produced from round green seeded plant will show the alleles capital R and small y, and wrinkled yellow seeded plant in its gamete small r and capital Y alleles are there. When they combine, when we hybridize these two in the F1 generation, the progenies genotype, or in the F1 generation all the progenies, their genotype is capital R small r, capital Y small y. That is their genotype. And what is their phenotype then? That is they will show round yellow seeds. All the progenies will show round yellow seeds. Then when we self-pollinate the F1 plants, that is now the parents are round yellow seeded plants into round yellow seeded plants, but they are heterozygous plants. When we self-pollinate or hybridize these plants, we can see that from one F1 plant, 4 types of gametes are produced. 4 types of gametes. Here you can see the first type of gametes are capital R, capital Y alleles. Gamete having capital R, capital Y allele, and the other gamete having capital R, small y allele, and small r, capital Y allele gamete, and the other gamete with small r, small y. Like that, 4 types of gametes are produced from male and female parent. And when these gametes combine to form progenies, we can represent it in the form of checkerboard. Here you can see how many gametes are produced here. 4 into 4. Okay, 4 from one parent and 4 from the other parent. When they combine, you can see that when they combine, how many progenies are produced here? Yes, 16 progenies are there. 16 progenies are there produced. From the 16 possible progenies, you can see the genotype is given here. You can also write the genotype yourself, and here the genotypes are given. And from this genotype, when you see the first one, capital R, capital Y allele, if it combines with capital R, capital Y allele wala gamete, then it forms the progeny. The progeny will show capital R, capital Y. Then what is the phenotype of the progeny? Yes, the progeny will show round yellow seeds. The progeny will show round yellow seeds. Like that, you can count how many progenies or how many offsprings are showing round yellow seeds. And then also you can count how many progenies are showing round green seeds. And then you try to count the number of progenies showing wrinkled yellow seeds. Number of progenies showing wrinkled yellow seeds. Wrinkled yellow means they will show small r and capital Y combination. Capital Y small y or capital Y capital I. Then they will be showing wrinkled yellow seeds. And here you can see that the last only one progeny, it is showing wrinkled green phenotype. And when we count, we will get this ratio. Here the parents were round green plants were crossed with wrinkled yellow. In the F1 generation, we got all the plants were round yellow. And in the second generation, we got this type of combination. Round yellow plants are there, 9 round yellow plants. When we count, we will get 9 plants are there. And round green plants, 3 plants are there. And wrinkled yellow, 3 plants are there. And wrinkled green, only 1 plant is there. Then we can say that the dihybrid F2 phenotypic ratio is 9:3:3:1. Here we can see parental combinations are round green and wrinkled yellow seeded plants. In addition to that, the first and last one, that is round yellow seeded plants and wrinkled green seeded plants are also produced. These are the recombinants or new combinations produced from this dihybrid cross. In the F2 generation, these are the new combinations produced. Okay, here when capital R, small y was crossed with small r, capital Y, in F1 generation, all were capital R, capital Y, small y, that is round and yellow seeded plants. When self-pollination of F1 plants gave parental type of plants and two mixtures or two recombinant type of plants. What are the recombinant type of plants? Round yellow seeded plants and wrinkled green seeded plants. In the ratio, these parental and recombinant progenies are produced in the ratio 9:3:3:1. The conclusions from dihybrid F2 ratio, the conclusions are round and yellow seeds are dominant characters. The occurrence of new phenotype combinations show that genes for round and yellow seeds are inherited independently of each other. Occurrence of new phenotype combinations show that genes for round and yellow seeds are inherited independently of each other.